For this activity, we are identifying water as a constituent in food. The materials required are meat, eggshells, plant leaves, and crucibles. First, we grind and crush the meat, eggshells, and plant leaves in their dried form separately. Next, we put them separately into the crucibles and heat them. During heating, we hold a glass sheet directly above the crucible. Finally, we use anhydrous cobalt chloride, or copper sulfate, to identify whether the liquid drops that form on the glass are actually water. When we test it, we can observe that blue anhydrous cobalt chloride paper turns pink, and white anhydrous copper sulfate powder turns blue. This confirms our conclusion that water is a fundamental constituent of living tissues. Activity 3, which is the identification of the presence of carbon in biomolecules. The materials required for this are several crucibles, spinach stems, a piece of fish, and chickpeas.
We begin by placing the ground pulps. Strong heating leaves behind only the black carbon residue, which rubs off easily on paper.
Test. Once it is cooled, the final residue obtained should be rubbed against a white paper. Observations. For our observations, black carbon lines are drawn on the paper. Conclusion. Because lines drawn due to coal are observed, we can confirm the food that is used for the experiment contains carbon. In conclusion, biomolecules contain carbon as a core element. The purpose of this activity is to figure out if a specific food sample contains starch, which is a complex carbohydrate plants use to store energy. What we are doing, step by step. Preparation.
First, grind the food sample with water. Filter the mixture into a clean test tube. Add a few drops of iodine solution.
When iodine comes into contact with starch, it chemically interacts with the starch molecules. This reaction instantly changes the color of the solution from yellowish-brown to a dark purplish-blue or blue-black. The conclusion. If the liquid turns that distinct blue-black color, it acts as absolute proof that starch is present in the food sample. We are conducting a food test to identify the presence of sucrose. If we add Benedict's solution directly to a sucrose sample and heat it, there will be no color change. Instead, we must add a few drops of diluted sulfuric acid to a freshly prepared sugar solution and heat it first. After this step, we add a few drops of Benedict's solution to the mixture. Next, we immerse the test tube in a boiling water bath and heat it. Watch the solution closely as the chemical reaction accelerates. As it heats, a distinct series of color changes will appear. The solution transitions from blue to a green-yellow, then orange, and finally it forms a brick-red precipitate. This final brick-red color is the exact confirmation we are looking for. Our conclusion is that sucrose is present in the sample. We are going to identify the presence of simple carbohydrates by conducting the food test for reducing sugars, like glucose. To begin, we obtain a liquid food sample, in this case, a solution of glucose, in a clean test tube. Next, we add a few drops of Benedict's solution to our sample. You will notice it starts off with a bright blue color. For the chemical reaction to occur, we need to add thermal energy. We immerse our test tube into a boiling water bath and heat it up. Keep a close eye on the liquid as the temperature rises. Observations. As it heats, a distinct series of color changes will appear. The solution shifts from its original blue to green, then to a green-yellow, turns orange, and finally settles into a thick brick-red precipitate. Conclusion. This final brick-red color is the ultimate chemical proof we are looking for. Our conclusion is that reducing sugars, like glucose, are indeed present in the sample. To start our experiment, we need a food sample. We can use a filtered solution made by grinding dal, or simply use an egg white and place it into a clean test tube. Next, we add a solution of sodium hydroxide, or NaOH, to our sample to create the right alkaline environment. Now for the crucial step. We follow up the sodium hydroxide by carefully adding a few drops of copper sulfate, or CuSO4, solution into the test tube. Watch closely as the chemicals interact with the molecules in our food sample. As the reaction takes place, you will see a rapid and beautiful color change. The mixture transforms entirely, turning into a distinct purple or violet color. This vibrant purple or violet shift is the exact visual confirmation we are looking for. Our conclusion is clear: protein is definitely present in the sample. To start, we add a liquid lipid sample, such as a small amount of coconut oil or gingerly oil, into a clean test tube. Next, we carefully add our chemical indicator, the Sudan-3 reagent, directly into the sample. Sudan-3 is a specialized, fat-soluble dye, which means it binds directly to the oil rather than mixing with water. As the mixture settles, you will observe a very distinct reaction. Bright red fat globules will appear and float right at the top of the test tube. Conclusion. This separated bright red floating layer is our visual proof. Our conclusion is that lipids are indeed present in the sample.
To begin the experiment, we obtain a filtered food sample, such as a solution made by grinding dal or using an egg white, and place it into a test tube. Next, we add a solution of sodium hydroxide, or NaOH, directly to the food sample. Once the sample is prepped, this step is followed by carefully adding a few drops of copper sulfate, or CuSO4, solution into the test tube. Watch closely as the chemical indicators mix with the biological sample. Observations. As the reaction takes place, you will witness a rapid and beautiful visual shift. The mixture transforms completely, turning a distinct purple or violet color. Conclusion. This vibrant purple color is the exact chemical signature required for this test. Our conclusion is absolute. Protein is present in the sample.